So I'm here to talk about our Enhancing Nutrient Use Efficiency in Cotton project. So notice I said nutrient use efficiency, and the title I should have really changed from more profit from nitrogen to more profit from nitrogen in brackets and phosphorus, because very early on in the stage as the project was developing, we were asked to include an emerging project to do with phosphorus. And part of that reason is why you see the massive team that we have here, and in terms of roles, sorry, I've thought about putting pictures in, but couldn't figure out where to put them. We sort of have the researchers team, and then the massive research support team, technical officers, technical assistants, casual assistants, and lots of farm help at the Australian Cotton Research Institute. And then nearer to the bottom of the screen there, you can see the team that we had at University of Queensland and the University of Melbourne, and also the University of New England, UNE, supporting postgraduate research as part of the program. And we also have Nylantha and James, not part of the official team, but working together with us in some collaboration. So the objectives and aims of the research: increase our understanding of soil and fertiliser N&P use in high-yielding irrigated cotton systems. We wanted to investigate the possible interactive effects of fertiliser placement, timing, and even though it wasn't part of our official brief, we threw in some enhanced efficiency fertiliser experiments as well, irrigation scheduling, and all those effects on how it affected nitrogen use efficiency and improved plant phosphorus nutrition. Yeah, and the aim was to come up with some improved soil crop and irrigation management strategies for the industry. So we had a whole range of research questions, and I've taken the approach of listing each one and just picking out some highlights from each one rather than sort of discussing the whole thing. We had 5 nitrogen ones and 4 phosphorus ones, so I'm going to do my best to get through them fairly quickly. So the first one for the nitrogen was looking at the current status and of what was known about nitrogen fertiliser management in the cotton systems. We basically did a review, I guess, and look at research, published research, and when you look at published research, you really the mass of that came from the late Ian Rochester, formerly based at Narrabri at Cotton Research Institute with CSIRO. So he published a lot of his data over the years. So there was a big core of data from him, and also industry survey data was looked at, and some of the key outcomes from that review, which turned into a paper. As you can see in that chart there, and this is a chart of some on-farm rate trials by Cotton Info and CSD, Cotton Seed Distributors, on-farm variety trials. So a large range of amounts of fertiliser being applied, but you can see if you draw a line through that, it pretty much goes across the page. So I guess the key take-home was that above about 200, 240 kgs of nitrogen per hectare, we're just not often seeing increasing lint yields. So there's other factors limiting yield, and in brackets, the take-home was more N won't necessarily fix it, so don't keep applying more N, I guess. But we thought that from the work that had been done, so that nitrogen use efficiency gains could be made by improving some of the management options like timing and budgeting and accounting for residual soil and mineralisation. Question number 2 was done in collaboration with the University of Melbourne, looking at the significance of ammonia volatilisation as a nitrogen loss pathway in furrow irrigated systems, and furrow irrigation is one of the key irrigation forms. It's not the only one, but it's one of the main ones practiced by the cotton industry in Australia. So a specific case study was done over one season in Moree using a slant path, I think it's open path, Fourier transfer infrared technology. Probably didn't get that right, but Dr. May Bay from Moree came up and ran that equipment with help from other colleagues at Melbourne, and looked at the emissions of ammonia coming out of the field over 3 in-crop nitrogen applications. I guess the key finding from that was one of the common ways of applying nitrogen in-crop is to broadcast urea, then follow up with an irrigation within a day or so. We found there was pretty much very minimal ammonia volatilisation from that. But one of the other, perhaps less commonly practiced but still out there in the industry, practices of applying ammonia directly injected as a gas into the irrigation water. So fertigation was producing, we measured 24% of that N was leaving the paddock as ammonia following that practice. And I guess the other thing, you can see it in the graphs on that page there, the water running through with the ammonia injected into it. A lot of that's being fairly unevenly applied across the paddock. So the concentration of the ammonia in the water drops with distance from where it's been injected, so towards the end of the paddock, but did increase over time of the irrigation. Question number 3 for nitrogen was, how does the interaction of irrigation management and nitrogen fertiliser timing practices affect end-use efficiency in cotton? So this is, I guess, one of the bigger ticket research things in our project, and as you can see in the photo there, it took the place of large field trials. So the plots in these trials were 8 metres wide by between 1 and 200 metres long. So we basically took up entire paddocks at the Australian Cotton Research Institute, where we had these looking at interactions between irrigation frequencies and nitrogen split timings. It was over 2 seasons, and we had 2 irrigation frequencies, so a low frequency of irrigation, a high frequency of irrigation, and somewhere between 3 or 5, depending on which season it was, fertiliser split timings. So I guess the key take home from a lot of that work was that there was really no interaction between those 2 strategies that we found in terms of cotton yields. We did find that the irrigation frequency, which is shown on the graph here as a deficit, so there's the 2 colours in the graph, there's a 50 mil deficit and a 70 mil. So 70 mils means that basically the soil gets drier before you irrigate it, so there's a longer period between irrigations. But sometimes what happens when there's a longer period is that it gets drier, so you have to put a lot more water in, and that can have a consequence in terms of yield as well. So we found a deficit effect, but it wasn't really wasn't always a consistent one. It really interacted with the type of season that we had. So we did have deficit treatments in all 4 seasons of our field work, but only 2 where we're interacting with the timing here. And where we looked at timing, which we did over 4 seasons, but not always in interaction with these, we did find some timing effects, and you can see there was a timing effect in this data here, which was from 2017-18. And in this particular example, shown on the graph, the 100-0, which meant that 100% of the fertiliser was applied pre-plant, it actually yielded lower than where either some or most of the fertiliser was applied in the crop rather than pre-plant. That season, we had other seasons where it was effectively reversed, so where 100% was applied in crop in the 2019-20 season, we actually got the lowest yield from that treatment. So we think it's mainly to do with the interaction of what type of season that we had when we were applying those treatments. So in this 2017-18 season, it was fairly dry in terms of rainfall, whereas it was totally the opposite with the 1920 season. So we got a lot of excessive vegetative growth at the expense of lint production. But another interesting fact from this piece of data here from 2017-18, if you look at the 100-0 2 bars there on the left, so these treatments were applied without extra phosphorus fertiliser. So we weren't starting off by thinking that this was a paddock that we're going to have a phosphorus response, but since the project was to do with phosphorus, we needed to be looking at potential for phosphorus responses, and we were actually surprised to see that we got a significant response to added P in this site, which, when you look at the soil test, did not have a low Colwell P in terms of what's recommended for industry as a deficient site. So we got this response, and then that sort of led us to, and we also got a response in one of Gunnar Natchamuthu's other project trials, and so that sort of led to more investigation that I'll come to in some of those other research question slides. But in terms of timing, just one other fact there, that there was, or has been, some increasing interest by farmers in, anecdotally, that a late application of N, late in the season that is, so in sort of February, usually the nitrogen applications in crop finish around January, but a late application in February had some farmers thinking that was increasing their yield, so we put that in as a treatment in our final year's trial, and we also looked at it in an on-farm case study. in our trial we actually found that it actually decreased yield in the plus phosphorus plots and made no difference in the nil phosphorus plots. So it wasn't a winner in that 1 year of experimenting. Question 4 of 5, nitrogen. So how does the method of in-crop? So we looked at in-crop methods, products, and sort of more detail of the methods, I suppose, again through field experiments at ACRI. Here we looked at broadcast versus side dressed, so drilling the fertiliser beside the plant in crop, versus fertigation, so basically dissolving the urea and running it with the irrigation water. As you can see in the photograph there, we had different plots set up with the products in those shuttles that were pumped in with the irrigation water. And we did find that in terms of the nitrogen loss from the paddock, there were differences between the treatments, but it didn't impact on yield. The losses we found were greater, and these are cumulative runoff end losses over a whole season, were greater from the water run or the fertigation treatment than from the broadcast or the side dressed treatments in 2 seasons. When we looked at fertigation and compared different treatments within fertigation, we had UAN, ammonia and urea. We found that the loss cumulative over the season was greatest from the urea. It was sort of equal to the ammonia in the first season and equal to the UAN in the second season, but I guess keep in the back of your mind that the ammonia one also has that extra 24% loss from, or potentially from the volatilisation directly as ammonia. So you can basically double the size of that bar for the ammonia treatment. And then in terms of, what can we do about it in terms of the water run urea, in the last 2 seasons you can see there we had the standard treatment and what we called an optimised treatment. The optimised one was basically shutting off the nitrogen supply into the water at the time when the water starts running out of the other end of the field. And to irrigate these fields, you have to run the water out of the field for sometimes many hours while the water in the field is continuing to wet up the plant beds. Number 5 of 5, how does the irrigation management affect organic matter mineralisation of N? And we looked at this over several seasons. Again, we had the 2 more and less frequently irrigated treatments there. You can see in the graphs there's 50 mil and 70 in the first graph and 60 and 90 in the second graph. And again, the higher number means it's watered less frequently. And so what we found basically with that was the more frequent watering, so more wetting and drying cycles during the season produced more mineralisation. But I guess what we noticed when we were doing this, and it did affect the yield, so these were in nil application of nitrogen plots, and we also found that the mineralisation was really concentrated in the centre of the plant bed, underneath where the actual plants are, and it's much reduced out in the furrows and sort of in between on the hillsides. But what we found when we were trying to measure this is that we're trying to measure mineral N being produced, and it moves with water through the plant bed, and that makes it kind of hard with traditional methods to measure. We tried applying a bromide tracer as well, and so it's not behaving or thought to behave similar to nitrate in the soil in terms of movement, but not so much in terms of plant uptake. And it showed that, what we were measuring in those top graphs was potentially only half of what was being produced. And some modelling done more recently supports that. So the actual amounts of nitrogen being mineralised through the season could be well over 100 kgs of N per hectare. And the bottom graph in this doesn't really fit with the mineralisation, because it's actually a graph from John Baird's PhD work where he's looking at 15N nitrogen applied as fertiliser, and you can see the application point there. It's just and if you want to see the detail of this, watch his performance in the Soils Conference coming up. But what he's really showed is that the nitrogen moves quite readily and rapidly through the plant bed and out towards the side of the bed opposite from where the water from the irrigation is coming. So it's moving laterally across and out into the furrow and exiting the paddock, or some of it exiting the paddock. So on to the phosphorus research questions, 4 of these. So the first one was looking at, is there long-term stratification of phosphorus or declining soil phosphorus in cotton farming systems? So to do this, we looked at a bunch of archived soil samples from 5 multi-year cotton farming system experiments. And we looked at the samples that were taken at the start of the long-term experiment and those at the end, and where possible we went back to those same paddocks and sampled them again now, because some of them had finished several years beforehand, or perhaps more than several, a number of years beforehand, and also some native vegetation sites. So I guess a couple of the key findings of this study was, out of those 5 sites, the 3 things that we were measuring in terms of phosphorus, solution P, Colwell P and BSES P. The BSES P more of a, I guess, background reserve, including background reserve phosphorus as well as the more active components. So all types of phosphorus that we measured declined in those locations, except for the one from the Macquarie Valley, which was really low to start with and had been fertilised during that particular experiment to increase it. And you can see that actually on the bottom graph on this page. So it's increased over time from 1994, the first sample was taken, 2001, the end of that trial, and then 2018 when it was sampled as part of this project. So a fertiliser plan there has rebuilt it, but it's being rebuilt in the surface, obviously where a lot of the fertiliser is being applied, where the plant residues are breaking down as well. In terms of the top graph there, which is from one of the long-term experiments at ACRI, you can see the decline there has been, we think, tempered a little bit by the crop rotation of maize that's been introduced, and Gunnar's I guess hypothesis is that this is bringing the maize into the rotation has helped improve the soil structure, making more. So the actual overall pool of BSES P has declined more than the Colwell P. So more of what P is left is available to the crop. So that's certainly provided some interesting data there. Number 2 of the phosphorus questions is how much has been taken up from the cotton plants from the surface and subsurface layers. And again, we've looked at those archive sample results to come up with the results that are presented here. But there's also been a PhD study program with the University of Queensland running as well, when they've specifically looked at some of this in that program in terms of where the P has been located, but I haven't got that here to show at this time. But the graph that we're seeing here is again from those archive samples. You can see that in terms of BSES and Colwell P, there's a contribution from different depths is 37 to 42% near the surface, about a third coming from that next 15 centimetres and then sort of tailing off below that, and very little from below 45 centimetres. So number 3 of the phosphorus questions, how is the response of P being affected by varied irrigation and nitrogen management? So as I said, we started bringing in plus and minus P as a very simple way of looking at this question in 2017-18, and that's where we found that quite interesting response, which the one I showed you before in the circle is in the top left of this diagram on the top here, and you can see that where we've got nitrogen optimised, where we've got phosphorus optimised, and where we've got the irrigation system in that season optimised, we got standout yield, and where everything was not so optimised, the yield dropped at the other end of that graph. Pardon me. So based on that, we started looking at okay, well, how can we get the phosphorus into the system in a more strategic way, I suppose. So we've not only had these N by P by I experiments, but we've looked at the plus and minus P in the N timing experiments and PIX, which is a sort of a growth regulant experiment, and our N rates that we've had as well. Even the water run treatments, so we put everything plus and minus P in the final season. And there was also, as part of the UQ program, looking at fertiliser of P uptake under varying water supply regimes. There's some glasshouse work there that's still being written up. So the key findings, I guess, from this, that both N and irrigation management really influence P response, as we said, in that low in-crop rainfall season. So I guess addressing that with applying we say more P, so having some P, some nitrogen, up front, really helped improve the situation and the P response, potentially due to this increase in early root exploration, and one of our other postgraduate studies is looking at roots specifically related to phosphorus, and some of that data is in the bottom graph here, where again you can see with that nitrogen applied in the 2018-19 frequently irrigated, which was dry at the start of the season, then got very wet. That really optimised the root uptake. The last one of the phosphorus RQs was looking at P movement in the water runoff. So all of the water runoff sampling that we did throughout the 4 years, we looked at reactive and total P as well. And in the final season, we also looked at sediment P coming out. But we also looked at samples that have been collected from Guna's long-term tillage and rotation trial there. And one of the findings from that, you can see in the middle there, the maximum tillage, there was less, I guess, overall phosphorus net runoff, I suppose, because we're measuring runoff, we're measuring input as well. So we're looking at the balance, and the balance was basically telling us that the more phosphorus was ending up in the paddock than was going away. And that was more the case in the maximum till than the minimum till, where you can see there was a couple of years where there was more running out of the paddock than came in. And a couple of interesting ones that probably not of great agronomic use, but interesting to us, that the application of nitrogen fertiliser actually reduced the amount of P in the runoff water, which was interesting. But we're not talking huge amounts here. To sum up our industry knowledge and understanding outcomes, basically, if we want to improve fertiliser nitrogen use efficiency, yes, we've got to either improve yield or decrease nitrogen fertiliser. Because a lot of the yields aren't limited by nitrogen, we've got to address these other factors: potassium, acidity, compaction, whatever they are. And until then, basically the nitrogen rates, people need to look at reducing them if they want to improve nitrogen use efficiency. Otherwise, a lot of that applied nitrogen is either lost, exported in the seed, so the seed N concentrations we measured, and some of those increased with a lot of the N that was applied, or we find it in the soil at the end of the season. We think you really need to budget the nitrogen rates to include that existing soil N, and trusting the soil test is a bit of an issue for people in the industry at the moment. Nitrogen fertiliser timing, enhanced efficiency fertilisers, irrigation scheduling all reduced the losses that we were measuring, especially in terms of runoff. Fertigation losses, we found a fairly, I think, easy method to avoid. To reduce those by turning off the fertiliser supply when the runoff starts, or avoid them by perhaps not using injected ammonia. And in terms of phosphorus, we found that the soil P is declining over time and stratifies when you try and rebuild it. Our cotton soil and plant management that enhances our P acquisition by improving the root growth. And our responses can be seen in these non-critical co-LP soils. Likely, if we can get that early root exploration going through adequate early season N, that doesn't necessarily mean you need to apply it all up front, but we need some applied up front. And in some of the spin-off experiments, we looked at how to apply it, and actually applying it to a preceding cover crop was actually gave a better response in the following cotton crop than applying it to the cotton crop directly. We got basically better distribution of the P. And generally more P enters the paddock than what leaves in the runoff. So the project outputs, Marguerite's talked about these a lot already. We've got papers, manuals and conference presentations, etc. Thank you very much.